Good morning, everyone, or good evening, wherever you are. Thank you so much for being here. Welcome to our Gene Delivery Seminar Series for the month of February. I am very excited to have today's speaker, Dr. James Allen. Dr. James Allen is an HHMI Hannah Gray Fellow and incoming faculty at Mayo Clinic at Massachusetts General Hospital and Harvard Medical School. He received his PhD in molecular genetics and genomics at Washington University, working on melanocyte stem cell generation using the zebrafish model system. During graduate school, James was a recipient of the HHMI Gilliam Fellowship and as a postdoc, the Burroughs Wellcome Postdoctoral Enrichment Fellowship. As a postdoc, he trained at Massachusetts General Hospital and Harvard Medical School, and his future lab will use zebrafish models to study how pediatric cancers like acute lymphoblastic leukemia co-opt genetic pathways to drive malignant transformations, advance the disease, and resist therapies. Thank you so much, James. We are very excited to hear you talk about your research. I just want to let everybody know that this session is recorded, and if you have any questions, there is a little Q&A box down below. Put your questions anytime, and they will be answered at the end. With that, over to James.
All right, thank you for that introduction. So I guess I'll just jump right in. Well, first, thank you to Vector Builder for inviting me. This is a really great opportunity. And I just wanted to mention that one of the reasons that I was really excited about this is that a lot of my research really uses plasmid-based experimentation in our design. So Vector Builder is a company that I could see myself working with a lot in the future. But anyway, I'll just jump right into it. So I usually like to start with a very basic question that drives me in my research. So that is, what causes cancer? So there's this idea that normal development, usually in the context of the hematopoietic system, is leading to the development of immune cells. And so the idea is that what I'm interested in is the idea of cancer initiation, where in some process in the normal development of these important cells becomes defective, and that leads to malignant transformation. And the reason why this question, what causes cancer, is somewhat open in many contexts is that it requires trackable systems, which usually involves preclinical modeling. And often these are driven by heterogeneous combinations of drivers, which requires you to also model the complexity that's seen in the patient population. But even beyond that, we know that different malignancies can develop from the same cell type. So, for example, we can have different types of subtypes of the same disease or leukemia that develop from the same cell. So, how does that happen? And this can actually have very real consequences for the patient. So, for example, there's two subtypes of acute lymphoblastic leukemia characterized by the presence of what's called the Philadelphia chromosome. And so Philadelphia positive ALL tends to have a better prognosis than another subtype called Philadelphia-like, which doesn't have the translocation, which tends to have a bad prognosis. So different leukemia subtypes can have very different outcomes for the patient. And so it's really imperative that we understand how these different presentations of the disease develop so that we can better treat them and help the patients. Um, as I mentioned, I study acute lymphoblastic leukemia. So you might be wondering what that is. So this is essentially an overgrowth of lymphocytes, which are the precursors to T and B cells. So this is caused by a developmental arrest of the immune cell lineages. And so what I'm showing you on the left here is a normal smear of human blood. So you can see that it's mostly red blood cells. But in a patient with ALL, for example, you would see the presence of blast in the periphery, indicating that they have grown out of the bone marrow of the thymus and have entered the bloodstream and are now just over proliferating and accumulating within the body. And so they come in two different broad subtypes based on their cell of origin. So we either call them B-ALL if they come from a B cell, or T-ALL if they come from a T cell. Sorry if I said that incorrectly. So this is actually one of the more famously success stories in cancer therapies, I would say. So one of the things that really drew me to study this disease is that, over the course of decades and a lot of research from various groups, this has really become a very treatable disease in many regards. So I'm showing you here just a tracking of the various trials that were performed by St. Jude. Uh, Dating back from the 1960s into more recently until 2017. So what we can see here is that over the past decades, the 5-year survival of these patients has remarkably improved. So this used to essentially be a very almost like a death sentence, not to be crass about it, but now patients that are diagnosed with ALL with the onset of targeted therapies can actually expect to have a pretty good survival if they are treated with the disease. Um, what has remained true, unfortunately, and this it creates something of a clinical paradox, is that in cases of relapse, so in the case of a patient has achieved a remission of the disease and some years have gone by and then the disease represents itself. Unfortunately, this happens in about 15 to 20% of these patients, and at that point, the therapies are not as effective. So, for example, most of the drugs that are used for frontline therapies are just not responsive in patients when they experience a relapse. And so, for that reason, it's really critical that we understand, factors that drive relapse, things that drive the disease, so we can better understand and treat these patients that are dealing with this ongoing risk of relapse throughout their lifetime. And so, to add to the complexity of this, I mentioned that there are two broad flavors, T-ALL and B-ALL. But even beyond that, there's actually almost 40 molecular subtypes of this disease. So each dot that I'm showing you here, this is a massive genomic study performed by groups at St. Jude, uh, basically just showing that when they took all these patients and sequenced them, molecularly characterized them, 1,300 patients and nearly 2,000 BALL patients, they were able to characterize them or organize them into different molecular subtypes. And each of these subtypes has a different prognostic factor, different responses to therapies, and different outcomes. So this is really just there's a growing appreciation that there's more complexity in the patient population that we initially thought, not just a simple characterization of B or T, for example, or the cell of origin. There's some really truly sophisticated and complex biology going on underlying these patients that we're all broadly characterizing as ALL. And so that requires a refinement and more research and understanding, for example, like what drives these different subtypes and how can we better treat each of them as they present in the clinic. So, just to step back from my perspective, what my interest in. So, like I said, the normal immune cell development happens where you have a lymphoblast, which is a precursor, and then ideally it would make a mature immune cell or T or B in the body. And so, what I like to think of it is that the how a leukemia can occur is that there's a lot of different roads, if you will, that can lead to this occurrence. So, for example, you can have chromosomal translocations, the formation of fusion genes that drive oncogenic transformation within those cells. You can have aberrant transcriptional signaling. You can have epigenetic remodeling. You can have any of these individual events, or you can have a combination of all of them. And this is just naming a few. There's more here, but just to emphasize the point that there's complex molecular events underlying the onset of this disease. And furthermore, when you take into account that there are so many different subtypes of disease that are likely driven by independent or necessarily not necessarily the same events, it creates a lot of complexity in how you actually model and determine what causes these different types of cancers. And so here we come to the zebrafish, which is my main power horse at this point. So I really enjoy this system for a number of reasons, but I've just highlighted a few here. So as a model system, I think it's probably one of the best we can use because 70 to 80% of disease-causing genes in the human have a zebrafish ortholog. Independent of that, as you'll see in the studies that I'm showing you here, the evolutionary conserved pathways present in zebrafish are so conserved that many of the human genes are actually oncogenic in the zebrafish. Which what I mean by that is that you can express a human gene in the zebrafish, such as KRAS, BRAF, any of the genes that we know cause cancer in the human, and you can actually give the zebrafish cancer using that. And I was struck by that because I thought that was a really powerful and system that you can really use to study these fundamental basic processes that underlie these malignant diseases. And it has a fully functional immune system, adaptive and innate. So they have T and B cells, for example, which is ideal for what I do. And they're ideal for high throughput in vivo discovery of cancer initiation, which is what I'm going to talk to you about. Okay, so I trained in at Mass General in the Langenau lab, who is a lab famous for studying relapse and progression in pediatric cancer. And here's where I learned how to do basically what I'm going to talk to you about today. So this lab uses innovative models of zebrafish leukemia to really get at the processes that underlie relapse and find therapies to better treat them, for example. So we developed zebrafish transgenic models of acute lymphoblastic leukemia. So I'm showing you here the blood smear like I showed you before. So by using thymic restricted expression of known ALL oncogenes such as c-Myc and Notch1, you can give the zebrafish leukemia. So I'm showing you here that you can directly visualize this with fluorescent reporters. So for example, by also expressing GFP, you can see the GFP positive cells, which are the transformed blast in the thymus here in the fish. So the upside down T is the thymus, sorry, just to explain it better. So basically, this is a fish that has leukemia because you can see the green has spread everywhere. This is a lethal condition for the fish, and it actually responds to standard treatment regimens such as radiation, chemotherapy, and targeted therapy. So this is a really useful model, I think, to study this disease and actually get at some of the fundamental questions that are hard to track in different experimental systems. So, and you might be asking why would you care? And not to be so forthcoming about it, but just because you can give a fish an oncogene and make it green, does that really matter? Well, it turns out if you do this and you design your experiments correctly, you can actually get at some fundamental properties of the human disease that you can discover using the zebrafish. So I'm just highlighting three studies that I was fortunate enough to participate in where we use the zebrafish leukemia models to discover this general theme of phosphorylation cascades that play a major role that are conserved across different species and actually are helping inform new treatment strategies for patients. Um, so anyway, so getting back to the patient population, so just focusing on TALL. So I mentioned that there are many subtypes of this disease, as I'm showing you here on this UMAP space. Each dot is a patient. But what you don't see here is that 60% of these dots or 60% of these patients have an activating mutation in Notch1. We've known this for a while. So this is we've actually known this before the onset of these different subtypes. So this actually creates something of an interesting paradox for me because you have all these different subtypes of TALL, but they are seemingly driven by a common dominant oncogene in the Notch pathway. So what that tells me, or at least that's what we thought when we started the study, was that one of the missing pieces here could be that maybe Notch is driving all these different subtypes of this disease because we don't know the other collaborating events that are capable of collaborating with Notch to initiate the different subtypes. So while Notch itself might be driving a few of these, maybe Notch plus something else is pushing it toward another one, for example. And so this was basically the onset of where the study began, trying to understand the collaborating events that might cause different presentations of these diseases. Um, so just to step back, just to talk about zebrafish, how you use the zebrafish system. So what I'm going to be talking about are transgenic zebrafish. And so there's a couple different strategies that the field has used quite robustly to generate transgenic zebrafish. The goal here is to deliver the material, which is usually plasmid DNA constructs, into integrated into the zebrafish genome so that you can achieve stable expression. And so, usually this is done, historically, this was first done with toll-2 and transposase. So, for example, I'm just showing you here that if you have a construct that has the toll-2, uh, uh, a toll-2 construct, then you will integrate it at multiple sites in the genome, sort of pepper them in, if you will, at discrete locations randomly in a stochastic manner. And so basically, you'll generate a lot of copies of this single construct across the genome. So that's how Toll2 works. And then there's also 5C 31 AdP based landing sites. So this is somewhat new, and I think this is really remarkable, actually. So this basically requires that you make a single integration site containing an AtP site, and then you use the AtB recombination to basically make it a landing site so that you get a discrete single copy of your construct. And this is very good for making stable lines. And if you want to learn more about that, I would encourage you to read the citation there I put in there because that's a really remarkable paper. That had nothing to do with. I just, game recognized game. Anyway, um, then linearized plasmid DNA, for example, is different. So this is a little bit more old school. So basically, this doesn't require any additional vehicle or accessory molecule. So, for example, the other two strategies are using integrase and transposase protein to actually achieve the integration. So, when you inject linearized plasmid DNA, which is just a circular plasmid that's been linearized with an enzyme, for example, you will basically create tandem plasmid copies at rare integration sites. So this is not good if you're trying to make like a stable line because it's very unlikely to um, go to make a stable line. But we decided to use this strategy for the screen that we had in mind. I'm going to show you why now. So the idea is that when you inject linearized DNA, as I said, you can create concatenamers that are basically just translocations at a point in the genome. So basically, the linearized DNA is integrating, and when it does, it's basically coming in as tandem copies. So if you just inject like a single construct, like the one I showed you on the last page, UBGFP, then you would just make a bunch of tandem copies of UBGFP. However, I reasoned that if you inject a complex pool containing a lot of different constructs, then you'll create a tandem concatenator containing a lot of different copies of constructs, different ones, expressing different genes, for example. And these will tend to co-segregate together. And so basically, by doing this strategy, you can look for combinations or combinations of genes that are capable of initiating a cancer just based on how they integrate into the cell. Um, so the constructs that I use in the study, because I pulled this up with the Vector B software just quickly. It was very easy to use. Basically every construct that I'm going to show you in the study has the same design on purpose. So basically, this is a zebrafish RAG2 promoter, which I showed you before to drive thymic restricted expression. And then driving a gene adventure. So in this case, either mCherry, which is our marker, or whatever gene that is listed there. So all of the constructs are essentially the same. They are linearized with either NOT1 or XO1, and then mixed in the pool and then injected, just so you understand the rest of this. So like I mentioned, the lab I worked in had done these sort of transgenic strategies before to identify collaborating oncogenes. So I'm showing you a study that we published some years ago where we basically were using this linearized DNA injection strategy to identify genes that collaborate with c-Myc, a known driver of leukemia, and basically looking for genes that, in combination with MYC, make the cancer or the zebrafish leukemia more aggressive. So, just showing you the results here, so that if you inject MYC by itself, for example, this little red line here, you get like an onset that looks like that. But if you combine it with any number of factors that were identified on the screen, you actually make the cancer more aggressive, and that we interpret as a collaborating event because it collaborates with c-Myc to make the cancer grow faster and grow in the fish faster. So what I wanted to do instead of this is that I wanted to, and instead of looking for genes that collaborate with known oncogenes, I wanted to look for genes that synergize with each other to initiate the cancer. What I mean by that is that because c-Myc itself makes cancers completely capable of making cancer, you're looking for an acceleration of the c-Myc oncogenesis event. I wanted to look for genes that gene A doesn't make a cancer, gene B doesn't make a cancer, but only when A plus B are together, you make a cancer. And so I use the linearized DNA injection strategy by making a complex pool of candidate genes at low concentrations of each. And then I linearize them all, place them in a pool for my injection mix, and then injected them into the fish. And so the idea here was that, as I said, when you inject linearized DNA, it tends to integrate as concatenamers. And so the logic here was that because the DNA is going to integrate, the different copies will be carried with each other, and only in the case where genes that collaborate with each other are carried together and co-segregating within the genome, I should detect the onset of cancer, basically. Yeah, so for example, in this case, so I'm showing you here one of the ones that worked. If this fish had integrated from the pool ICN set and LMO2, then I should detect if that fish gets cancer. I would infer that something in that fish that integrated into that fish caused the cancer, for example. And the key here is that individually, all of these genes do not cause leukemia in the fish. And so that was the logic here. And that's how we designed the study, just to look for if we could detect collaborating events that synergize in order to initiate this type of cancer in the zebrafish. And so this worked pretty well, actually. So I'm showing you an example at the top here of a normal fish that is our background strain that doesn't have cancer. So you can see the thymus label with the upside down T and GFP. And so you can see here just this is an H&E stained section of the tissue. So this is just basically just showing you that this is what a normal fish looks like from the gills and the muscle and the blood smear that we took from the fish. Uh, in contrast, this is a fish that we injected with what we call pool 1 plus notch. That's ICN. That's just annotation for notch. So basically, you can see here that not only is the fish covered with red blasts based on the fluorescent image, you can see here on H&E staining that their blasts just completely infiltrating and the gills and the muscles, and then the blood smear that we took from it contained blasts like we expected. So this basically told us that something within this pool 1 was capable of synergizing to initiate this cancer. And so we did this. We recovered many different type fish from this experiment. So we injected the single mix and just collected fish 1, fish 2 until we had a lot of them. And then we performed bulk RNA sequencing on each subtype or on each sample in the study. And so what we were expecting, because we were using genes that we associated with T cell leukemia, we were expecting to get mostly or only T cell models. But what we actually found was that when we looked at them, it seemed that we were actually creating multiple subtypes of both T and B. So on the left here, as I'm showing you here, the yellow pool here above here is the MYC model I showed you before that we had used in the lab, and those are basically the T cell models that we had seen before. But basically, what I'm showing you here is that the different injection strategies we did. So we took some genes out, we added some in, we kind of played with the pool a little bit just to see if we could actually change the dynamics that we were seeing in our screening strategy. And basically, we were able to recover multiple subtypes of T and B leukemia, which is pretty remarkable because we didn't actually think that would be possible with this strategy. Um, and so then this left us with a problem which might occur to people listening to this, which is that we injected these DNA constructs that are driving these oncogenes in the fish, and we got a tumor. We don't know, we still have the problem that we don't know what actually caused the tumor or to form. And so because we had done all this bulk RNA-seq, And because we had designed all our plasmids the same way, we were basically able to use the bulk RNA-seq to identify the transgene specific expression of those individual orthologs. So what I mean by that is that we were able to identify the reads that uniquely map to the transgene and not to the endogenous zebrafish genome. And so this is basically just showing you a display of the calls we made on saying, for example, this fish expressed the LMO2 transgene that was expressed from the RAG2 promoter in the construct we introduced experimentally, and not the endogenous zebrafish LMO2 ortholog, for example. We were able to filter those out and distinguish them, basically, and things I don't have time to get into. But basically, what we noticed is that we basically looked for the transgenes that were expressed in each of the individual samples. As I showed you before, pool one only has 10 constructs in it. And so we basically found that we were getting high integration rates of most of the pool, either because the concatenamers are quite large or there's multiple independent integrations within those samples. Hard to say. But what we noticed is that because we had done so many, our logic was that, for example, if we look at these green samples here, even though we were getting some samples that were getting high integration rates, and by the way, those tended to be the early ones that got the cancer faster and probably would have died sooner. Because we had collected enough samples, eventually we were able to find samples that we inferred that. So, for example, even though these early ones had essentially all of the pool in them, when we sequenced enough of them, we were able to find a sample that only had these three: Notch, Elmo2, and Set. And so we inferred that because all of these samples have this combination, and we found at least one sample that only had those three, we inferred that those three genes were the minimal combination necessary to form that leukemia, for example. And so we called it that group. And so we use that strategy by just sequencing many, many samples to find the minimal combinations necessary in the synergy strategy to identify potentially to nominate combinations that might drive tumor onset in this pool strategy. And so we were able to basically say that because we initially were looking for mostly Notch driven because Notch is the main oncogenic driver of TALL. But we were able to find both Notch synergies, like I'm showing you in green, and Notch independent, meaning that Notch was not even injected, but we were still able to get leukemias models. So that was pretty cool. And so the next thing we had to do was we wanted to validate them. So what I mean by that is like we nominated these combinations. What happens in theory, if we're right, if we inject just those genes without the rest of the pool, we should get the cancer as well. And so that's what basically happens. So I'm showing you three combinations that we were able to parse down. We did a lot of different injections, but we whittled it down to just sets of dual pairs, if you will. And so I'm basically just showing you that we were able to identify three combinations that robustly generated leukemia. So Notch plus LMO2, Notch plus SET, and IL-7 plus SET. So SET, which we didn't know going into this, actually was a dominant oncogenic driver in our screen, and we're uh, basically saying that SET is a dominant oncogenic driver of ALL initiation because it's capable of initiating with both with both Notch and other pathways that we know are oncogenic, like IL-7 receptor, for example. So what is SET? SET is highly expressed in ALL patients. So we kind of we're not necessarily surprised by this, but we are the first to report a direct role of SET in ALL initiation. So, and then here I'm just showing you that we didn't just get lucky on these combinations. We tried a lot of different ones, and I'm just highlighting two that didn't work at all. So yeah, so basically the strategy required that we inject a lot of different plasmid DNA constructs. We whittled it down so that we could nominate combinations that we think are present across enough samples to make them a likely candidate for validation. And we were lucky that, fortunately, that they did validate, and we were able to recreate we were able to validate them as actual drivers of ALL initiation. Yeah. So SET basically is a multifunctional protein. It does a lot of different things, and from transcriptional regulation to histone chaperone to inhibition of protein protease 2. So really highlights the role of epigenetic regulation in leukemia onset, which is something I'm really interested in looking for in the future. But anyway, so this was a really cool validated step. So we when we found this, we were very happy with this because it said like, okay, this strategy worked. We were able to start with a group of genes and we were able to whittle it down to identify like a known a new collaboration that no one knew just based on this study. And so, as I mentioned before, SET is highly expressed in ALL patients and BNT, so it kind of makes sense. Like it's just a robustly overexpressed gene, and so it likely has a pretty big role in them. Um, so anyway, so we did that based on the 10 genes that I showed you before, and because that worked, we decided to up this to 64 genes because we had all the constructs and we just wanted to see if this would work as well. And so basically, we employed the same strategy, but instead of 10 genes, we basically made a injection mix of 64 constructs at low concentrations and then injected them into the zebrafish, as I said before. And so this worked out pretty well, actually. This was actually a little bit more oncogenic than the first pool. Probably, but only with the Notch collaboration. It actually turns out probably we're diluting whatever is oncogenic in pool two, so we didn't get as many. But that's just like a detail. I don't mean to make a big point of that. So basically, what I'm showing you here is that we got a robust onset of tumors with this pool two plus ICN strategy. So I'm just showing you here the different factors that we include in our screen and what they're broadly characterized at. And on the bottom here, when we finally had all our samples, there are 155 individual screen fish samples in total collected from the study. We did principal component analysis and basically were able to determine at least 8 different clusters of samples in our study, which was really remarkable. We didn't really think this would happen. So not only were we getting different subtypes of T and B, we were getting genetic, molecularly distinct subtypes, clusters of samples, which is really cool because no one's ever done this before, at least in the fish. And so, and what's really remarkable is that this G8 here I'm showing you here, this the all the MIC samples, the MIC leukemia samples that I showed you were the model we'd used before, all clustered together. The rest of the clusters are all the ones that we generated in the study. And then I'm just showing you here the breakdown by whether or not we call them as T or B and what age they were when they were collected. So anyway, and this expanded study not only just kept giving us leukemias, which most of these fish in the study were, but it actually remarkably yielded these unique presentations of cancers that we had no idea what to make of. So, like we had these weird fin growths that were solid, and actually many of them, the one in the upper left here, we kept cutting it off and it would grow back, which was unexpected. We still don't actually know what to make of that. And so I'm just basically pointing out here that, like we expanded the screen and we got these not only leukemias, which we got a lot of, we got a lot of different things that we didn't expect, which is actually one of the cool parts of doing a screen because you never really know what you're going to get if it's designed well enough. Um, so anyway, the different subtypes, the clusters that we got were differ in aggression. So, for example, some of the clusters, the G, were less aggressive than others, and they seem to be driven by different combinations of transgenes, which was also pretty cool because it kind of independently confirms what we had initially observed in our first screening strategy. So, for example, G1 seems to be driven by a combination of Notch, IL-7, and LML2, whereas G4 was driven by IL-7, LML2, and Tau-1, and not so much Notch. Yeah, so it told us that we were getting different cancer models that look different. Molecularly, they appear different. Pathologically, they look different, and they seem to be driven by different combinations of transgenes, which is really cool. And then, probably the most surprising result of this study is that I'm showing you on the left here all the samples in the study based on and showing you the T and B enrichment that we used to call tier or B. So all the clusters at the top, the lineages that we called, and then we use NMF to identify novel genetic programs that are active in each of these subtypes. And so they're actually different because they're turning on different programs that I'll talk about in a little bit. But surprisingly, when we looked at our transgene calls across these different clusters, we found that for what for whatever reason, we're not still exactly sure why, the dominant drivers of this study were still a combination of ICN, SET, and IL-7, which are the combinations that I validated in the first study. We were not expecting that at all. So this was quite remarkable. I will say that this wasn't really a case of honeypotting the mix because in the second strategy, each of these genes was present at a very low concentration, equal to the rest of the pool. So it basically told us that like we were really only getting, and I should say that when we injected these fish, we had to inject a lot of embryos to get these numbers up. Like I think on the order of 20 to 30,000 injections. And then we tracked over 500, 600 fish individually to get these numbers. So it basically just told us that, so we had a lot of negative is what I'm getting at here. So it basically told us that like for some reason we needed some combination of these genes to really drive most of these cancers, which was pretty remarkable because it says that this is really it reinforces the idea that SET is actually a dominant oncogene across these models. And it helped us to identify some of the things that are going on in these cancers. So, like I mentioned, we did NMF to identify enrichment of programs that are active in these subtypes. And so I'm just labeling a couple of programs here on the side here, program 1, 2, and 4. And as I mentioned, the G8 was a RAG2 MYC model. So it's really active in the MYC program, which wasn't a surprise. But what's really interesting is that we were independently able to make another cluster that drives the MYC program without the MYC oncogene being introduced. And so that was really cool because it basically said that like, well, this cluster is really aggressive and it makes sense, but it's actually turning on MYC without actually having to have MYC there. So it's kind of converging on it. And this basically led us to use the enrichment of these programs to analyze, the patient databases to see if we could learn anything from the patients based on the models that we had made in the fish. And so basically, we use this MYC program we called a P4, and we're able to use the hazard ratio to determine that MYC is actually a good predictor of outcomes in TALL. So I'm showing you here that it enriches for MYC targets, so it's a bona fide MYC list. And basically, then we use these zebrafish gene signatures to ask, is the human, would a list of the human orthologs of these gene signatures in the fish, does that actually teach us anything about the patient? And so basically what I'm showing you here is that when we use a combination of the G6 program, or the G6 subtype and the P4 program, which G6 is activating P4, so we sort of like overlap them together, we basically show that TALL patients here, we can actually stratify the patients and predict outcome based on the expression of these zebrafish signatures. And this actually worked really well in a couple different subtypes. So on the left here in D, I'm showing you all of the samples in the study. So this is the 1,300 patients that I mentioned before. But if you just focus on a couple different subtypes, particularly ETP-like, you can actually predict event-free survival for the entire study based on the expression of this G6 low P4 high gene module, which we thought was pretty cool, actually. So we basically use the fish and we seem to be hitting at something that's actually present in the patient as well, and got there independently using a screening strategy. And so I'll just start concluding. So the takeaway message is that we use this strategy to discover that SET is a dominant driver of ALL. We identified 11 unique models of zebrafish ALL that we've never observed before, and that these novel models will allow future investigations into drug discovery and cancer initiation. And I hope I've convinced you that the zebrafish is a high throughput system for cancer initiation. So A couple of different things here. So like one of the things that I mentioned before is that I'm really interested in like looking at what molecular factors drive cancer initiation and the pre-leukemic state. So moving forward from the study, I'll be really interested in using this and similar strategies to really get at, this idea of like, what factors are actually driving the lymphoblast to become a leukemic transformation, or what pre-epigenetic states are actually making these cells susceptible to malignant transformation, for example. And then, beyond that, there's a lot you can do with this data set that we've made. Like I said, like we had 155 samples, and all of them have been preserved in the freezer, actually. So we have samples of every single one for the most part. But, there's a couple different things you can do from this. So, for example, I didn't talk about this, but G2 and G4 were basically excluded from a lot of our analysis because we found that they had a high level of immune cell infiltration. So for whatever reason, they were seemed to be contaminated, if you will, by a lot of T, B, NK macrophages, neutrophils, which basically means that these clusters, which are cancer because they did transplant in our assays, have an ability to really engage the zebrafish immune system in a way the other models don't. And so this could be a really powerful system to really look at immune cell interactions with cancers that we're potentially looking into. Another idea is that, like I mentioned, all of these samples are frozen viable, so we can do like transplant-based assays to do drug discovery. So, for example, we could, maybe in one of these different models that we have made, in addition to being like a G4 cluster, it might actually be resistant to frontline therapies or targeted therapies, which would be really cool because it would give us another avenue to study what some of the factors that clinically present challenges for these patients. And then also, of course, because we've identified, these collaborations, we can make interchangeable intercrosses with stable lines to make infinite number of models for these studies, so we don't have to do like the pool-based integration to randomly get these transgenes in there. And so I just want to leave you with this idea. This is my final slide. So uh, this broadly applicable strategy, I think, of doing mosaic screens with linearized plasmid DNA, it's not necessarily that any of these strategies, like I mentioned, like TOL2 or 5C, none of them are bad. I don't really think of anything in that way. It's just what is most useful for the question that you're wanting to ask, I would say. And so, you can imagine this. I think this is a very useful strategy if you want to do any cancer discovery in the zebrafish. So, for example, you can modify the screen with a CRISPR system to inactivate or activate genes. Like, for example, we had thought about doing this for the study, but we didn't get to it. So we had actually ordered this construct from Vector Builder, funny enough, to get dead Cas9 to couple that with the pool to see if we can activate some other interactions to make the cancers more aggressive in using the CRISPR system. We had also thought about new genes, so we had 120 candidates ready to go for B cell leukemia to start the strategy over with a B cell mindset and see if we could discover even more models of these leukemias. But I just want to emphasize that like my interest is in leukemia, and it probably will remain there for the rest of my career. But this strategy could be used to discover any cancer model you want. So it only really requires you have a good promoter that drives robust expression in your tissue of interest, that you have a good list of candidate genes, and it helps if you have some evidence that mosaic expression of these constructs is sufficient for malignant transformation. So I can see this working very well in AML, rhabdomyosarcoma, liposarcoma, melanoma, just to name a few. So yeah, I hope that this has been informative on some way, because if you're familiar with zebrafish, this is somewhat unorthodox, I would say, as a strategy, but I think it works pretty well. So anyway, I just want to thank you for your attention and happy to answer any questions.
All right. Thank you so much, James. That was absolutely wonderful. And also, thank you for including specifically what experiments you used the vector builder vectors with. It's always great to know exactly how we helped. So that
Yeah, yeah,
was
yeah.
wonderful. Okay, so we have a couple of questions. I'll get started with this one. Is there any viral system that can be used for stable integration of the GOI in zebrafish?
I'm not familiar with a viral system that works that well. I would love if that actually did work, because obviously that would be easier than doing all the manual injections. I do think one thing that could be done is that, I've thought a lot about uh, doing like integrated system where like you maybe start a larger like because I only did like 10 to 100 genes, which is like because we had a pretty good idea that they were going to work. But you can imagine doing like a genome-wide screen of some sort in cell culture using a viral system, and then whittle that down to your candidate list. Any hits from that, then moving it into the in vivo system. So that's probably the strategy I would employ in the future, using the viral integration at that stage, where it works really well, rather than necessarily bringing it to the zebrafish model, I would say.
Thank you. We have another question. I was curious about any new biology that might have come out of this work. For example, in the c-Myc program that was driven without c-Myc, if you looked at the genes expressed, does this make sense with what we know topologically, linking to this program regulation? Or is there a missing link suggesting a gap in our current topological knowledge of these expression programs and pathways?
This is a really good question. I really thank you for that question because this is actually what we're currently doing in our revised manuscript now. So essentially, we've I don't want to give too much away, but we've revised our thinking about this a little bit, and so we actually are thinking that these programs that we discovered are actually more they do hint at some novel undiscovered biology that we might have hit on. Just because it doesn't hit up in the database doesn't mean it's not relevant, for example. So I think what we've done here is that some of these models are actually hitting at a maybe undiscovered pathway that we're trying to characterize right now. And that will be really interesting to show in the future. So thank you for that question. I think that's where we're going to head on that. So yeah.
Awesome. Our next question is, are there specific safe harbor sites used for integration in zebrafish?
Um, There weren't, but the paper I alluded to, the 5C from Christian Moiseman's lab, has created the piglet lines, which are really remarkable. So their lab has done the laborious work of integrating landing sites for at P using the Toll2 system, and then uh, laboriously locate mapping the location. And so those are probably what the field should use in the future. Again, I had nothing to do with that. I just really like that lab. So yes, so more like comparable to what Rosa 26 and the mice. I think looking forward, the idea of doing these random Toll2 integrations might be, it's not that's going to go away, but I would love the field to move toward more using mappable integration sites, like you mentioned. So to answer your question, yes, there are available from that lab, and I think he's made more. Yeah, so yeah, they're there, and I would love to work with them actually. So I'm hoping to get them.
Wonderful. What is the benefit of mosaic screening versus barcoded screening?
Yeah, it's a good question. Well, I think well, for one thing, th this is probably a fault of a remnant of how labs used to work. So a barcode would have made my life a lot easier. I'll be the first to admit that. All this cleverness that may have come across or may not in this talk would have been completely welcome and welcomed removed if we just had a barcode in these backbones, which would have made the screening process a lot easier. I don't think there's any necessary real difference. I think moving forward, I would use a barcode to identify to as a necessary part of the screening feature. As it happens, this strategy was basically using constructs that had been made in our lab before, and so we didn't really have to do that. But if you're going to start from scratch, you might as well barcode them, I would say. I don't think there's any advantage or disadvantage to the mosaics per se. The mosaics are more powerful if you have a barcode. I'll say it that way.
Cool. We have a technical question, kind of.
Okay.
For the recombinase integration strategy, do you use existing sites in the zebrafish genome or do you integrate at the ATT site?
I guess it I guess for Phi C, you would integrate at whatever landing site you're using. For Toll2, you don't need any knowledge of that because it just integrates wherever. It just grabs the Toll2 sites and shoves it in the genome, essentially. It's interesting. Technically speaking, we talked about this. You could have done this same strategy without the linearized strategy. You could have done it with Toll2 because they'll just integrate together. They'll on some level they should at some frequency they should co-segregate together as well. It just seemed a little bit more potentially toxic, I guess. But it's slight preference for me to do the linearized concatenamers, I guess. But technically speaking, from a technical standpoint, you could have done the same if you wanted to do this similar screening strategy to discover genes that cause cancer. You can use Toll2, and you don't have to know anything about a landing site or anything to do that, I would say.
All right. I guess we can close with one like broad question. With the screening strategy, the strategies that you have developed, which is so cool. What kind of broader biological questions do you hope to answer or could theoretically be answered with this?
Yeah, that's a good question. So, one of the things I specifically would like to get at are, it's not just this cancer initiation is this broad topic, because obviously every cancer has to initiate, but I think, you can really get at more like specific clinical presentations of diseases that might be not as well studied based on how we do research. You could also look at, like I said, I'd really be interested in coupling this with CRISPR because I would like to look for cancer vulnerabilities. Not just the idea that you can start the cancer, but like discovering things that, if you knock out this gene, for example, the cancer will stop in its tracks. That would be very useful. I think that would lead us to these pathways and sort of get at this sort of maybe hierarchical understanding of how these pathways are activated or co-opted by these cancers throughout the progression of their disease in meaningful ways so that we can actually understand, like, given a snapshot of a cancer as the patient shows up to the clinic, where are you at in the disease progression, even if we don't necessarily look at it that way. Like, does it turn on this pathway and then suddenly when it gets to a certain stage, it turns it off, for example? So I think there's a lot of cool ideas that you could get at using this sort of strategy to just to understand like how things start and what happens as they move forward. Yeah.
Wonderful. There is one more question. Has there been loss of function studies for the zebrafish oncogenes? Is there a Cas9 stable line? It would be convenient for the drug screening, etc.
Yeah, there's a couple of Cas9 stable lines that I have found, or at least from what I understand from my colleagues, is that for some reason zebrafish do not like to express Cas9 all the time. So a lot of people have used like a mosaic strategy or honestly injecting the protein and just to achieve like the excision event early on. But there's been some studies that have used construct-based Cas9 delivery, pretty remarkable to some success to discover cancer biology related to that. So I'm thinking of like the Zahn lab has done this really well, and that's probably something you could look into if you want to learn more about that.
Awesome. And I think with that, we will end our February talk for the Gene Delivery Seminar Series. Thank you so much, James. This was really terrific. And thank you for taking the time and sharing your research with us. And thanks everybody for attending. We hope to see you in March or April or one of our future talks.